So my name is Brooke Edmonds, and I'm with OSU Extension, and I've been coordinating the Master Gardener Advanced Training webinars. So I have a little bio I'm going to read on Melodie, and then we'll pass it over to her. So Melodie Putnam has been with Oregon State University as an Extension plant pathologist and Chief Plant Disease Diagnostician since 1993. Her main responsibilities are to provide diagnosis and management recommendations for all of the samples submitted to the OSU Plant Disease Clinic. The last statistic I could find was from 2005 and showed that Melodie has personally diagnosed more than 19,000 plant samples. So I'm sure in this last couple of years you added quite a few more to that. So I'm really excited to have Melodie come and join us and do this dive into plant galls, kind of a neat thing that we see out there in our Master Gardener world. So we're going to follow the usual format, like Mark said, and then in a day or so, I'll be sending out an email with a link to the recording and a survey for you to fill out. And so with that, we'll just give a moment here and let this transition and let Melodie go through and share her screen and unmute and take it away.
Well, welcome everybody. I'm so glad you can join us, and I hope you won't be disappointed. But I do happen to feel that plant galls are pretty weird and wonderful things. And yeah, so if you're ready, let's do it. Okay, so it's always good to start with an understanding of what we're talking about. And in the broadest sense, a gall is just an abnormal swelling. But generally, it's understood to mean an abnormal swelling that is incited by some sort of organism that benefits from the gall. And galls can be formed by insects, by aerified mites, by fungi, or by bacteria. However, not all gall-like things are due to an organism. So for example, this is a sequoia that has these massive growths at the end of its twigs, and they occurred only where there had been some roadside spraying for weed control. I tried and tried and tried to find some sort of organism associated with these and never was able to. So if something is called a gall, it doesn't necessarily indicate what the cause is, just that some hormonal plant thing is out of whack. Okay. Page down.
Melody, sometimes you have to click on the screen on top to get your keyboard engaged. So.
All right, there we go. Okay, sorry. So a lot of people think that burls are incited by some sort of organism, and they are, but they're incited by the tree. So burls are generally an overgrowth of an area where there had been a branch of some sort. And you can see there was sort of a branch here. And sometimes they'll form where branches have fallen off. And why there's such a massive overgrowth, we really don't know. But burls can be pretty large. Here's one that's about the size of this bicycle here. But, you know, they're not harmful to the tree. They create problems if you're trying to grow timber and you want nice straight boards, but they're also highly valued for the interesting wood grain that they have on the inside. Another thing that looks sort of gallish occurs on apples and crab apples. These are called Berber Knots, and that's what I'm showing here. And these, again, are incited by the tree itself. There are lots of little root initials, and here you can see some little rootlets growing out. It's not really known why these occur. Some rootstocks tend to be more susceptible than others, but again, it's nothing pathogenic. And then once when I saw this new landscape planting on campus, I saw this little beauty and I thought, oh, that is so cool. They got galls on it. And then the person I was walking with said, those aren't galls, you dope. That's part of the plant. But still, you know, I thought it was way cool that they have these plants that form these structures that look like galls, and it's considered an ornamental. How cool is that? So anyway, not all things that look gallish are necessarily caused by organisms. So now, with that brief tour, I have some questions for you. So pick one, either A, B, or C, and they're not actually questions, but what the heck, we're all friends, right? So just indicate A, B, or C, and we'll see what the consensus is.
Okay, I've launched that poll. We'll just take about 10 seconds, folks.
Okay.
All right, folks are still voting, so we'll just give it another 5 seconds.
All righty.
But I think we have a consensus.
Oh, I can't wait to find out what it is.
Can you see those, Melanie?
No.
You cannot. Okay.
No.
So it was over, and this happens from time to time because you're in presentation mode. You probably can't see it. So I'll just, for the other polls, I'll indicate to you what the answer was. But yeah, the overwhelming favorite, 97%, was it makes total sense to me because not all things appear.
Oh, good. Okay. Because sometimes when I've done Master Gardener training in the past, people get really irate with me when I tell them or I show them things that aren't necessarily the topic that I'm supposed to be discussing. So good. And the reason I have this little graphic here is because as you stare at it, it sort of looks like this thing is turning, but it's not. It's just totally static. It's an optical illusion. And just a little reminder, not all things appear as they seem. So good. You guys are all on board. Super. All right. So my area of specialty is bacterial galls. And so that's what we're going to talk about first because I'm giving the webinar after all. So this is a plant that has this huge gall that was like almost the size of my head. And this was caused by the organism called Agar bacterium. And there are a number of different species that cause galls, but in general, a number of species of agrobacterium that cause galls, but I'm just going to refer to them as agrobacterium in general. And the disease they cause is called Crown Gall. And here you can see a massive gall. It's at the crown of the plant. It's at the base of the stem near where the roots come out. The Crown Gall bacteria have a huge host range. There are literally hundreds of different species that have been shown to be susceptible. And because I work a lot with nurseries, I see primarily herbaceous and woody ornamentals, but I've also seen vegetables affected. Primarily the plants that are affected are dicots, though, not so much the monocots or the grasses. Okay, I had to show you this little graphic because it's a nice representation of why or how Crown Gall occurs. So we have this little guy here, which is the agrobacterium cell. It's the bacterial cell, and this is a plant cell. And within the agrobacterium cell is some DNA, which is on an extra chromosomal piece of DNA. It's not part of the chromosome, it's outside of it. But it contains this little section here. And it has multiple genes along here, but we're going to focus on two regions here. One is the virulence region, and these are genes that initiate the response that results in disease. And this occurs when plants that have been wounded or otherwise injured produce phenolics and sugars that are in response to the wound. It's sort of a way to help heal the wound. And these are called inducers. And these inducers are these little green things, and they pass into the bacteria. They're detected by the bacteria. And they stimulate this virulence region, the genes involved in the virulence response. And that, in turn, initiates this movement of a piece of DNA that belongs to the bacteria, and it goes through the bacterial cell, into the plant cell, into the nucleus of the plant cell, and then becomes incorporated into the host genome. And as a result of being incorporated there, the plant starts making certain amino acids that are favorable to the bacterium. and also produce cytokinins and other plant hormones that reduce in the result in the massive overgrowth that we see with crown gall. And although it's called crown gall, it's sort of a remnant of where it was first found, where it was first noticed, but really the galls can form anywhere. This is a little, and I mean little, it is only, you know, a couple inches tall, lobelia that was produced as an unrooted cutting, and where it had been snipped before, a gall formed. And again, it was because the bacteria entered the wound, wounded tissue produced those inducers that were attractive to the bacteria and allowed it to initiate the pathogenic process. So the galls can occur anywhere. And what they consist of are just masses of unorganized plant tissue. So this is in response to being transformed by the bacteria. And here you can see a stem, a normal stem tissue that I've cut through the gall that was formed on the side of the stem. And you can see the vascular tissue here, this sort of ring here, is pretty well organized. But then once we get into the gall tissue, the tissue is very, very unorganized. It just is a massive proliferation of undifferentiated growth, which is very typical of crown gall. So here's 3 different images of leucanthemum or Shasta daisy, which is super susceptible to crown gall. And you can see that here on your right, there's a pretty large gall in comparison to the rest of the plant that's at the base. Here is a series of leaf galls that were formed when the leaf was wounded. Again, wounding is very important for crown gall initiation. And then again, we have a little gall down here at the base. So the thing to remember about crown gall is it doesn't always occur at the crowns. Oh, and here's a carrot. Oh, this was a beauty that came into the clinic just recently. Carrots have natural openings. Well, all plants have natural openings, but they have areas here called lenticels, and that's where gas exchange occurs in the soil. And Agrobacterium is considered to be a soil-borne bacterium, and it resides in the soil and can enter through these natural openings. And everywhere there is a lenticel, there was a gall. As I said, it can occur on woody plants as well as herbaceous plants. Here it is on a rose. Now, the thing about crown gall is that sometimes it is systemic in plants, but it's not uniformly distributed throughout plants in which it is systemic. So, for example, this rose, there was obviously some sort of wound here. And so the bacteria could be anywhere along this stem, or they might be elsewhere in these stems, but we don't know where because they're not uniformly distributed. And then once the bacterium moves inside the vascular tissue, it'll latch on to the xylem cells and it'll hold there really tightly and it won't be dislodged until something happens, and then the, you know, a wound or pruning cut or freezing damage or something that initiates those inducers that are produced by the plant, and then the bacteria will move out of the xylem, not out of the xylem, it'll move with the xylem up to where that wounding occurred, and then you'll get another gall. And they can be pretty honking huge. This is a Euonymus Gall, and you can see the size of the leaves in relation to the galls. Now, I suspect that crown gall is also systemic and euonymus, but it probably takes several years for a gall to get this size. So if this were a landscape plant, I would just prune it back here, and it might be another couple years before you see a gall of this size. And it's not really, I mean, you can hardly kill euonymus with a shovel. You know, it's not really injuring them to any extent. And then finally, there's another species of Agrobacterium that affects only grapes, Vitis vinifera, and it's called Agrobacterium vitis. And here there's a gall that has occurred at the grafting union. So normally, well, not normally, but often with grapes, you'll have a rootstock that's of one variety, and then the scion wood will be the desirable variety, in this case, Pinot Noir. And there was something that happened and allowed the bacteria to get in at the graft union, and then you got this nasty old gall. So this is one of those examples of a plant where the bacterium can be systemic, which is not very good if you're trying to grow grapes, because it is detrimental to grapes. All right. So what the heck do you do about crown gall? Well, there's no cures, okay? Once that transformation has occurred in the plant genome, there's no going back. So anything that's infected has to pretty much be destroyed. And I'm talking here pretty much commercial production, because it's a huge, huge problem in the floriculture industry and in many nursery industries that produce woody plant materials, especially apples and cherries and pears. They're all very susceptible. So when they get it, they can't really sell that material, because who wants to see something that looks so gnarly? You know, you're just not going to buy it. So they destroy it. There is an option of biological control, but it's limited in its effectiveness. It's another species of agrobacterium called Agrobacterium radiobacter, and it does not have the ability to cause galling. And there are two strains, the K84 and the K1026, which are formulated in commercial products. And these products work by this Agrobacterium radiobacter producing an antibiotic that is effective against some strains of Agrobacterium tumefaciens, the Crown Gall bacterium. It also works by just getting there first. So the way the product is used is as a pre-plant dip, for example, with cherry roots. The problem is that not all species, or not all strains of Agrobacterium tumefaciens are susceptible to this antibiotic. Some are and some aren't. So when it works really, really well. And when it doesn't work, it works less well. Okay. Then there's a product called Gallex, which is not a biological control. It's a xylene-containing product that kills the tissues that support the gall. So it's used primarily in walnut production, where walnut galls form on the trunk of the tree, and you paint that on and it kills the galls. But obviously it's not going to work in a situation where the bacteria are systemic. So one of the projects that I've been working on with the team here at Oregon State University is trying to find something that does work. And there hasn't been much research into applied aspects of Agrobacterium. There's been reams and reams and reams of information on Agrobacterium as an agent for plant transformation, but not simple things like how does it get around, how long does it persist, can you transmit it on cutting tools, and those are the sorts of questions we've been looking at. So one thing that we did, since this hadn't been done in a long time, is we looked at commercial products that are already on the market that are registered for bacteria control. And some of these are sanitizing agents. They're not all meant to be used on plants. Certainly you're not going to put bleach on plants. And these are a couple antibiotics. And so we were looking at the efficacy of these materials in multiple formats. One, we looked at how effective the product was on a solid medium where it diffuses into an agar and inhibits the bacterial growth. We were looking at an assay that uses a liquid medium. And then we were looking at the effect of these products on bacteria that are in a liquid suspension and those that are in biofilms. Biofilms are very cohesive and difficult to disrupt bacterial environments. It's sort of like a little bacterial city. So you don't have to look at the specifics of all of this. The green ones are the ones where they worked at the labeled rate. And we've since filled out these white areas. But in general, things only worked at really high rates. So that was a little disappointing. We tried also testing some of these products for Crown Gall control in plants in the greenhouse. And this is a typical result that we got. These are all of our controls. We had 10 plants each that we either wounded, because you need a wound, wounded and applied a buffer, and then treatment only to see if there is any adverse effect on the plant, which there shouldn't be since these are already labeled. And then this was our positive control, where we didn't put on a product, but we inoculated, because we wanted to show that yes, we do have pathogenic bacteria here. And then these are the products that we tested, and we applied them first and then applied Agar bacterium. Results are pretty dismal. I mean, there's no way. In some cases, it seemed to potentiate the size of the galls. Anyway, so that clearly wasn't working. So we thought there's got to be something that will get this. And so we were looking at various natural products, including plant extracts and bacterial extracts. Bacteria are known for producing lots of different antibiotics and other antimicrobials, so why not fight fire with fire? So we found this one natural product that is a result of some bacterial growth. And we tested it in the greenhouse on chrysanthemums, but what I'm showing you here are the tomato results. So what we did is we took a growing tomato, we cut off the stem, we applied the product, and then we applied the Agar bacterium. And this was the control where it was the product was not applied. This is just our pathogenic bacteria. And then here's what we got when we treated with the natural extract first and then inoculated with Agar bacterium. It's like, whoa, there's no galling at all. There's a little bit of a dieback at the top here, but we didn't see that with chrysanthemum, so that might be something specific with tomatoes. But this was really phenomenal, because we got the same result with chrysanthemums, and chrysanthemums are one of those super susceptible plants that are very problematic for commercial production, because it's all clonal. You know, they all just take cuttings from mother plants and ship those out, and if it's systemic in those mother plants, they might have transmitted the bacteria in those cuttings when they weren't showing symptoms yet. So this is pretty cool. So we're hoping to do some further work with this and maybe get it out as a commercial product one of these days. Don't hold your breath, though. These things usually take a long time. So stay tuned, but don't hold your breath. Okay. The other thing we were interested in is a detection system for nurseries, because they really want to be able to detect it quickly, and they want to detect it themselves. So on the left here is a commercial product, which is for a different bacterium. This is a Xanthomonas thing, and this is called a dipstick. And how it's used is it's a serological based assay, and you grind up your plant tissue and you put it in a little bag that's provided by the company up to this line, so that the sap goes up to here, and then you wait, and the sap will migrate through this porous filter paper-like material, and if this Xanthomonas is present, you'll get a strong band right here, and this band just indicates that the assay is actually working. Well, we thought that was pretty cool, but we wanted to adapt this technology for Agar bacterium, because it's not always easy to distinguish Agar bacterium from callus. I get samples from nurseries all the time with exactly that question. And we had to use a molecular assay. So these are the results of 2 different molecular probes that we were using to develop sort of a dipstick assay. And it works the same way. You have a control line at the top, and then if the pathogenic bacteria are present, you'll get a line down below. So here is where we wounded, and we just applied a buffer. There's no indication of detection. This is an avirulent or non-pathogenic, non-disease-causing strain of Agrobacterium. No detection. That's good. And all of these others are pathogenic or gall-inducing strains of Agrobacterium, and you'll see that there are lines. What this is showing here is that there was a difference in the ability to detect different isolates or different strains of the bacteria between the two probes. So probe 1 was more capable of detecting a wider range of strains than probe 2. And the reason we wanted to go to a DNA technology instead of a serological only technology is because you do have isolates of Agrobacterium that are in the environment that don't cause disease. So we don't want to detect those, because then, you know, growers would start throwing out healthy material. So this is an assay which is currently in development, and again, we hope to be able to make this available to the growers so they can do early detection on their own. Okay, so I'm going to switch gears now to another favorite of mine. It's called Rhodococcus. It's a little bit different distortion-causing bacterium than Agrobacterium, but what you're seeing here is a pea plant that has been inoculated with Rhodococcus and a normal pea plant. So you can see that the leaves are small and twisted and distorted. This has a very wide host range as well, and it infects similar sorts of plants as the Agrobacterium, but it can also affect monocots, which Agrobacterium cannot do. So Rhodococcus starts out as an epiphyte or a colonizer of leaves or sometimes of roots, and they'll just hang out on the surface for a while until there's some sort of triggering event, and we don't know what that event is yet. And when that event happens, that's why there's this empty box here. We don't know what it is. And the bacteria move into the plant and colonize different parts of it, and you'll get a different response depending on where the bacteria first land. If they land on the roots, then you'll get short, stunted roots. You won't have as much root hair development. The stem thickens relative to the healthy one, and then you get this formation of leafy galls. So when there's been a lot of research done on the molecular mechanisms of this bacterium, but not much else. So this is the surface of a pea root that has been inoculated, and we were interested in where the bacteria actually were, because they're only supposed to be in the first few cell layers, just a few cell layers deep. But if you look in this little box here, there's a dot right in the middle, and that's what this dot is. And it's an aggregate of cells. And if you look around, you see inside the cells, there are other bacteria there too. And these are the Rhodococcus bacteria. It's like, what the heck are they doing there? They're not supposed to be there. We don't know. So there's a whole lot of information that we still don't know about this bacterium. But we do know it causes plant distortions. And they're pretty dramatic. This is a pink, a Dianthus, with this mass of little tiny shoots. A similar leafy gall at the base of a leucanthemum. Again, the Shasta daisies are just as susceptible to Rhodococcus as they are to the Crown Gall bacterium. These are leaf galls that are forming on an Euonymus leaf. This is a healthy leaf, and this is one that's diseased. And then even on agave, you'll get these little growth distortions. And so a leafy gall is just a proliferation of buds that only partially expand, and they remain juvenile. They don't get any bigger. This was an image that was sent to me by a commercial producer, and you can see this huge honking leafy gall on this Petunia, if you can believe that. And you can see that it's really problematic for commercial producers. So again, like agrobacterium, there's no real cure. Sanitation is paramount. Again, it's extremely problematic for the producers, but there's really nothing that can be done in the landscape. So another quick gall question here. You could pick A, B, or C. Then we'll move on.
Okay, I've launched that poll, folks. We'll just take about 10 seconds.
So I'm going to be moving super fast through the rest of this, and it's because there's not as much known about many of the galls I'm going to be showing. But hopefully we'll wrap it up in maybe another 7 minutes.
Okay, so there is not unanimous agreement on this one. So
Okay.
70%, the majority was agrobacterium is super cool.
All right.
And then 15% for the other two
Okay.
responses.
All right, we're all on the same page. Excellent. Now, I'm going to, like I said, just sort of breeze through these insect galls. There are a lot of different species of insects that can produce galls. A lot of them are wasps or in the Hemipteran order, aphids and adelgids. Now, the thing about these insect galls is that they offer like a ready site for food, because this is an aphid gall, and the little aphids are wrapped inside this this distorted leaf, and so they've got a nice protected place to eat, and they're pretty much protected from environmental problems, although not necessarily from other predators. Here's some examples of galls that you're likely to see. This is a Cooley spruce gall, Adelgid gall. These tend to turn brown later in the season, and people think they have pine cones on their spruce trees, but they're caused by these little insects that go to the base of the needles and start feeding. And with all of these galls, there's some sort of plant hormonal activity that is stimulated that forms these strange growth. Here it is on noble fir. This is another adelgid that affects true firs only, but not Douglas firs. And you can see the amount of injury is relatively minor. Oak galls are particularly visible this time of year. This is called the spotted oak gall, sometimes called oak apples. They are caused by a little wasp that tends to lay her eggs on the veins, and it starts as just a little blip on the vein, and it grows larger and larger, and the little pupa, or the little larva, is encased in this little like chariot inside this gall that grows bigger and bigger. And here you can see the insect has crawled out. And there she is. She's stuck in these fibers. She says, I can't find my way out. But she's the little wasp that will then go on and eventually, after many adventures and forms, come back and lay eggs in the oaks. Now, there's a long history of information about these galls. These Aleppo Gall nuts are from a Mediterranean type tree. This is just a screenshot showing that you can still buy these galls commercially because they're used for ink, because they have lots of tannic acid in them. They're used for tanning and dyeing, and also for just about anything that ails you. You can fix it with one of these galls. So I've even seen it that it's good for hangnails, but I wouldn't put much credence on that. And remember, if you want to mark another celebration on your calendar, May 29th is Oak Apple Day. This is a celebration that occurs in places in Britain to celebrate Charles II's return to the throne after the overthrow by the Puritans and Oliver Cromwell. And there seems to be a difference in how the sexes celebrate this. Here, the women are very stately, and they're adorning themselves with apple galls, and I'm not sure what these guys are doing, but they're obviously having a good time. So however you choose to celebrate, May 29th is the day. Now here's, I said at the beginning, there's not a lot of information known. This is one of those galls that we found on an Oregon white oak that I just could not find any information about, and there was nobody home, so we couldn't figure it out. There are a number of swellings that occur on leaves and stems. This is a poplar gall caused by an aphid. And again, the little aphid is going to be tucked inside that gall and not really available for predation by birds, for example. And here's that kinnikinnick again. Here's a little ladybird. She's a ladybug aphid. She's looking for those aphids. Ladybug beetle. She knows those aphids are in there somewhere. She's trying to find them. This is a pretty spectacular looking gall because it turns pink as it ages. Then there are other things that are less common but kind of spectacular when you see them. The woolly elm aphid infects or affects the leaves while they are still developing, and the feeding activity and the saliva causes leaf distortion. Generally minor problem. Woolly apple aphid lives on the roots. They're sort of snowbirds. They go down and live on the roots during the wintertime, and in the summertime they'll migrate up and cause problems on the stems and sometimes the twigs. Here's one that you'll often see if you're looking about at all. It's caused by a little wasp that she lays her eggs at the tips of the meristematic tissue of the leaves or the twigs. And if you get one of these galls inside, open, you can see inside are these little larvae, snug as a bug in a rug. And you can see what sort of protection these galls afford these insects. So in landscape situations, there's really nothing that needs to be done. Some people just can't stand the sight of these things, so okay, just prune them out and get rid of them. But there's always a creative use you could come up with, I'm sure. All right, another poll. Select A, B, C, or D. Just give you an idea of whether you've ever seen them or not.
Okay, we'll just take 10 seconds. Alrighty. So the majority, although by a slim margin, is I've seen what I think are insect galls, and then number 2 was I see insect galls all the time.
Okay.
And just a handful of folks for the other options.
Okay, well, those of you who are taking medication, I hope it's working. All right, fungal galls. These are far less prevalent than insect galls, but you still might see them. There are specific galls that are very noticeable. One is this gall here called black knot, and I keep moving my cursor around because I lose it otherwise, so I hope it's not annoying you. Anyway, this occurs primarily on cherries, and choke cherries, and other members of the Prunus genus. It's a fungus, and this white stuff on the surface is the spores that are eventually produced on the surface, and then these are windblown or rain splashed to other twigs where they initiate more galls. This is a really serious problem in peach production. This is peach leaf curl, and this is an enlarged view of what it does. This is a microscopic fungus that produces spores right on the surface of these little swellings again. And as you can see, they can really hammer the trees. And in my area, if you're growing a peach, you have to use control for this organism. And then huitlacoche, this is corn smut. This is considered a delicacy in certain parts of Mexico. Here's some huitlacoche risotto. It's an interesting combination. This is where the fungus infects the developing grains and causes this massive growth. A lot of rust fungi will form galls. This is serviceberry with a gymnosporangium rust. This rust has a complex life cycle that transitions between members of the rose family and juniperous species. And here it is on the serviceberry. You can see all these beautiful orange spores that have emerged from the surface of these little fruiting bodies. And again, the galls can form anywhere. Here it is on the bottom of a leaf. And it can occur on fruit too, which is really problematic for the fruit growers who are trying to produce pears especially. There's one that you might see if you live in the Pacific Northwest and you're walking along the coast range where there's lots of huckleberries. This is another fungal swelling and a swelling of huckleberry, native huckleberry, that is caused by a rust fungus. And again, the rust has an alternate host of a fir. And here's another one that occurs in various parts of the country. This is called Western gall rust, or sometimes gall rust. It has no alternate host, but it occurs on 2-needle and 3-needle pines. And we see this both in the Coast Range in Oregon and on the other side of the Cascades in Eastern Oregon. But it occurs, gosh, I remember once I bought a Christmas tree in Maryland and it was covered with these galls. So it's not just an Oregon problem. All right, so for these galls, a lot of them, if they're in the landscape, you don't really need to do anything. But, you know, if you're in commercial fruit production and you really want those nice, big, fat, juicy pears, then you'll probably want to do some active management. And there's plenty of information available on how to manage specific galls. So I think because I've blathered on a lot, I'm going to end it here. I'm not going to go cover the Aryophyte mites, but basically they're Aryophyte mites are, I'll just do one. These are Aryophyte mites. They're microscopic mites that look different from regular mites, and they get into leaf tissue and cause little tiny disruptions. Like here it is on grape, and this is a magnified view. And so each one of these little things is a plant hair, and there's a little Aryophyte mite in it. And so they're nice and protected, and they cause these little swellings. But generally, they're not problematic for plants. I just wanted, here's some pictures. We're just going to breeze through these. We'll skip that one. Oh, here, this is where I wanted to get to. So here's a couple references that are available for more information about galls. This one's really nice. It has lots of color pictures. This one's also nice, but it's British, and so we don't have all the plants, and the information inside is from drawings. But they do have a key which allows you to key things out. Otherwise, you just sort of have to flip through the book and look through this one. And then one last shameless plug. I do have a Twitter feed called All Things Galls, where I like to post pictures. And that's available at my Twitter feed, which is at Oregon_Plant Clinic. So I'm going to shut up.
Great. Thanks, Melody. So we can't hear the clapping, so we'll clap for you. So if there's a couple questions in the Q&A, but if you have questions for Melody, pop them in there. We'll also pull those book references and get them out in the email to you as well, so you'll have a chance to look at this information. So I'll just start at the beginning for now. So you showed that picture in the beginning. I think it was agrobacterium and the carrot root. Mm-hmm. And so Jesse wants to know, could you eat that? Like, does it affect the flavor or the texture? I mean, it looks yucky, but if you have it in your home garden.
No, it's perfectly safe to eat. It's not a human health hazard. And if you just peel away that, the galls when you're peeling the carrot, it'd be fine to eat.
Oh, okay. So it doesn't go too deep. That's all right. Good to know. Meg has a question about going to the oak galls, so the ones on the leaves, the oak apples.
Mm-hmm.
And so she wants to know the wasp eggs, do they let bacteria in, and is that how that structure is forming, or is that solely really the wasp interacting with that?
Yeah, with all of the insect galls and the aerified mites, the activity of the organisms feeding is what stimulates the plant growth to produce that abnormal structure. So there's something in the buggy equivalent of saliva that causes the plant to produce those growths. So in the case of the oak apples, they're totally induced by the organism, and there are no bacteria involved.
Nancy has a question about roses. She showed a picture, I think it was like a feathery rose gall, I think. And she just is curious if anyone's looked at differences in susceptibility. I guess she's noticed some differences with wild roses and cultivated roses.
Yeah, that's not a well understood phenomenon because in my own yard I have lots of Oregon white oak seedlings that some of them are totally covered with oak galls and some of them don't have a single one. And I imagine it's probably the same with the mossy rose gall, is that there are just unknown factors that influence which plants are affected and which ones aren't. So there's not a lot of information available about insect galls, I have to say.
Yeah, it was kind of interesting to hear like just the gaps in the research. You know, you had slides with just there's a gap people don't know yet.
Yeah.
So yeah. Valerie, so you flipped through one of your slides about bright pink galls on a maple leaf. It was one of your last slides, and she's curious to know what that is.
Yeah, that's an aerified mite gall. It's called a bladder gall. And the little tiny mites are on the undersides of those galls, and they're encased in those little bladders. And the little bladders are filled, again, with plant hairs. And so the little aerified mites are all tucked away and cozy in those galls. I think they're quite striking. The aerified mite galls are often bright red, and they really catch your eye.
I have the ones on my grapevine. So yeah, could bring you lots of samples. Bridget wants to know if the bacteria that cause galls, are they at all related to human health, or are they strictly focused on plants?
No, they're strictly limited to plant infections, which is a good thing because as a researcher, I tend to be a little clumsy, and so I don't like working with bacteria that are serious to human health. So I stick with the plant pathogens.
Great.
But yeah, no fear, no transmission to humans.
Christine would like to know another rose question. If the roses have galls one year, are they likely to get them again the next year? And
I guess that depends on the environment. It's probably more likely that they will get them. I've seen plants that have them year after year, so I assume that they didn't all occur in 1 year, and I'm just seeing the infection from 1 year. I would imagine they would be.
Right.
No.
Are you putting in answers? So on the black knot, so the one on the fungal gall, it's strictly fungal caused, or are there any other organisms? I know some folks are from the Midwest. That's kind of a common one, I think, in the Midwest there.
It is more common in the Midwest and also on the East Coast. It's strictly fungal. And in that case, most of the tissue is more fungal than host material. But again, we don't really know why that swelling occurs. But it is very characteristic. It's one of those that once you see it, you know it, and you'll be able to identify it every time you see it.
Great. Were you looking through the questions? Did you have a comment on one of them, or no?
Me?
Okay.
Oh, no.
Okay. Jolene has a question somebody brought in, and they identified it as a horn gall, and it looks like antlers. And I'm not sure where Jolene is from. I don't know if you've seen anything like that. I think there's a lot of things that have like the orange horns. Yeah. So if you don't know it, that's okay. We can.
Yeah, it would be helpful to know what the plant was. If it was a rosaceous plant, for example.
Okay. Well, we might follow up with you on that one. A couple of these other ones are. Oh, Nancy wants to know what the biggest oak gall you've ever seen is.
Actually, the oak galls are pretty much the same size. I think the ones that I showed you were the largest I've seen. They tend to get smaller than that. It's just the bacterial galls that just keep growing and growing and growing.
Gotcha. Some of these other ones are a little bit, I think, maybe more specific. And so we'll be able to pull like a transcript of some of these questions, and we can, if it's something that we don't have the answer to, we might follow up with you on that.
Okay.
So I think we might leave it at that for now, because we're at our time, because some of these are more diagnostic type questions that we would want to follow up with where folks leave. So we want to say thank you to Melodie. We really appreciate you sort of showing us the inside of your world, and so we'll all follow you on Twitter now, right?
Thanks so much for letting me blather on. I could, obviously I could talk for a long time about this.
Then so everyone out there, if you need another credit for webinars for continuing education, our next webinar in the series is December 17th at 11 Pacific time, and it's diving into herbicides. So it's weed management in the garden and landscape, focused on understanding herbicides, so you can answer your questions as master gardeners. And I just want to thank you, and thank you, Mark. Appreciate it.
All right. Thank y'all.
Thanks.
Thank you. Bye-bye.